Welcome everybody, welcome to this edition of Monday Lunch Live. I'm very pleased to welcome you to this session, which is going to take us through quite a few interesting concepts related to this new CAR T-cell therapy. But before I do that, I'd like to open by acknowledging the traditional owners of this land, the Wurundjeri people of the Kulin Nation, and their elders, both past, present and emerging. My name's Dr. David Cock. I'm a radiation oncologist here at Peter MacCallum Cancer Centre, but I also work in an academic capacity convening the Master of Cancer Sciences for the VCCC. Most of you know that the VCCC is an alliance of 10 different partner organisations that's both hospitals, academic centres and research centres, and through combining the power of all of these, we really get to bring to the forefront some of the best oncology research and practice, not just in Victoria, but nationally and internationally. And today we have a great showcase of that. We're going to have a combination of two different presentations. So we're going to have Michael Dickinson, who's the lead of the Aggressive Lymphoma stream here at the Clinical Haematology Service at Peter MacCallum Cancer Centre and Royal Melbourne Hospital. He's also the principal investigator on several first-in-human trials on CAR T-cell therapy, and he's been very heavily involved in the setting up of the CAR T-cell service, which is recently been announced, funded here by the federal government, which is going to be a national centre of excellence in cellular immunotherapy. So we're going to hear a bit about the science and the professional side from Michael, but we also get to be really privileged to have Finnian Kenny, who's also joining us today. Finnian has actually been a first-hand recipient of CAR T therapy. His own experience was when he was diagnosed with double hit lymphoma. He'll be able to tell us a lot about this himself in much more detail than I will. So I'll leave that largely to him, but he's a great success story of this particular treatment and why it's a way that we're going, that this therapy is really opening up new avenues and really a way of the future. So it's a great pleasure to have both of them with us. We also have a great number of people who are online, who I'll just say hello to today. So there's, I think, 66 people joining us online today, which is one of our best attendances. And as part of this actual material is actually going to be recorded and used as a special lesson within the Master of Cancer Sciences as well. So a real great presentation today. I'm going to start by inviting Michael Dickinson up to the floor. Thanks very much for coming, Michael.
Hi everyone, so I'm Michael Dickinson, a haematologist here at Peter Mac and Royal Melbourne, and I run the work with the rest of the team on aggressive lymphomas particularly, but I also treat leukemia here. And I'm going to be talking a little bit about the science and the clinical data underlying this new class of therapies, and of course I'm delighted to have Finnian Kenny and Bernie, his wife, join us here today to talk a little bit about first-hand experience. This is Emily Whitehead, a photograph taken around 2012 at the time of her report in the New York Times and the New England Journal of Medicine, a young girl with acute lymphoblastic leukemia, now extremely famous worldwide because she was really the first case report of the incredible efficacy of this cellular immunotherapy. Emily had been refractory to all prior therapies, and at the time she was treated with CAR T-cells. She had very active leukemia in her bone marrow, also in her cerebrospinal fluid, as well as disease in some of her organs. And she went on to receive a treatment with this CAR T therapy that I'm going to be talking about today, which put her into a durable remission. And the report of that remission in the New England Journal of Medicine and the New York Times really hit a home run for this particular treatment, because anyone who treats acute leukemia who heard this story could see the potential for this particular mode of therapy. She's now, in fact, 9 years cancer-free and continues to tour the world talking about her particular experience. And although she wasn't the first patient to have a complete remission with this therapy, it was perhaps her particular experience that really convinced industry and researchers that this could be a way to beat hematologic malignancies. Today I'm going to be talking about CD19 positive malignancies. Not all, I'm going to be talking particularly on diffuse large B-cell lymphoma, but you can see that CD19 is a protein on the surface of B cells that's present from precursor B cells, which is where we see childhood leukemia and adult acute lymphoblastic leukemia arising, but also mature B cells, which are the normal correlate of some of our more mature B cell lymphomas, like diffuse large B-cell lymphoma, which is the most common follicular lymphoma, chronic lymphocytic leukemia, mantle cell lymphoma, and marginal zone lymphoma. These diseases all have CD19 on the surface of their cells, just like normal B cells through their range of maturation. And so for the rest of this talk, I'll be focusing particularly on this target for CAR T cells. There are many, many other targets for CAR T cells, but I'm focusing on this one and the clinical data here. So when we talk about cellular therapy and we talk about CAR T cells, we get excited about the idea of live cells being able to fight a cancer. But of course, the best example of this in clinical practice is allogeneic stem cell transplantation, a concept that many people will be familiar with. This is the idea that we can take a donor immune system and reinfuse it into a patient as a way of controlling leukemia or lymphoma or other diseases. And allogeneic stem cell transplantation was initially developed in animal models in the mid-60s, and subsequently patients were transfused with allogeneic transplantations in the late 70s, and then it became a very popular and then subsequently routine way of treating patients in the early 1980s, with now more than, I think something like more than 100,000 patients worldwide having been treated with allogeneic transplantation. So this is the idea where a patient's immune system is ablated with chemotherapy and then replaced by a matched donor. And it brings with it the potential advantage of controlling leukemia through mismatch of antigens on the surface of the leukemia cells, but the potential disadvantages of graft versus host disease. Graft versus host disease meaning that the donor immune system recognises the new host as being foreign and attacks it and causes a plethora of potential complications across pretty much any organ system. The graft leukemia effect in allogeneic stem cell transplantation is complex, and it's not just mediated by cytotoxic T lymphocytes. It's mediated by really many different arms of the immune system, including T helper cells. And it involves antigen presentation using the MHC class molecules to generate a response that targets the leukemia. And separating this graft versus leukemia effect from graft versus host disease, I would say is one of the key challenges in clinical practice today. But of course, we transplant patients and use allogeneic transplants to deliver cure routinely. And this idea, really is the first example that shows that the immune system can be harnessed to target hematologic malignancies. So you've heard the term CAR T. CAR refers to Chimeric Antigen Receptor. These are designer receptors that are then made to be expressed on the surface of lymphocytes, which are modified using a number of different manufacturing processes. Where CARs differ from the typical T-cell receptor is that the T-cell receptor requires multiple mechanisms to work. It generally recognises antigen only in the context of MHC molecules, and it requires co-signalling to be active and to activate cytotoxic T cells to kill their target. The interaction is low affinity, and so it's very difficult really to get optimum target killing out of simple druggable interactions with the T cell receptor. Where CAR Ts differ is that they are basically designed to harness target specificity and binding properties of antibodies, and also then use the cellular toxicity that comes with T lymphocytes. So we know that we have this humoral immune system where we make antibodies from B cells and we develop antibodies to target known antigens, and that antibodies can target their specific target very avidly and bind very avidly. And so what a CAR T does is it combines the target specificity of antibodies with the T-cell receptor, so that when you have the T-cell receptor now, this chimera, this hybrid T-cell receptor, has both the specificity of antibodies and the ability to activate T-cells. And there are multiple generations of CAR T-cells. We found that just simply creating an antibody specificity and binding it to the T-cell receptor was insufficient to lead to disease control in the human trials. And so further modifications were made to take some of the co-stimulatory molecules that are required to activate T-cells and incorporate the signaling mechanisms within the new CAR T. And there are now third-generation CAR Ts that have multiple co-stimulatory mechanisms. And the CAR Ts that I'm describing today generally have a single co-stimulatory molecule, and the ability of both binding and then significant T-cell activation through this co-stimulatory molecule was the key to the success of these second and third generation CAR Ts. And as I've mentioned, activation of the CAR T cell, or the cell bearing these CARs, does not require co-stimulation through these co-stimulatory molecules and HLA molecules, which is one of its key advantages. How do CAR T cells kill? Well, they kill through a range of mechanisms which are homologous to normal T-cell killing through release of granzyme and perforin upon activation when they see their target on the surface of tumour cells. They also express cytokines and expand and multiply when they see their target, and then go ahead and continue to kill cells exponentially. And they also seem to have an effect on the local milieu where the tumour cell is. The other thing that we've learned about CAR Ts is that they kill cells sequentially. So this is experimental data from here in Peter Mac, where you can see green is the T cell, the clear cell is the tumour target, and as they've said, red is dead. This is a marker that demonstrates that the cells have lost viability. So the green cells are the CAR Ts, which are going from target to target and sequentially killing their targets. And so we know that these T cells stay alive. They don't just kill one cell and move on. They don't just kill one cell and die, but they kill sequential cells in the tumour. So this is the other potential advantage of this designer immune system. So this is the manufacturing. And I'm not going to go into the detail behind the scenes manufacturing of genetic transduction and how it all works, but I wanted to talk to you a little bit about the basic process. Essentially, we have a target patient, and the idea here is genetically modifying their own lymphocytes. And so the first step there is collection of lymphocytes using leukapheresis, and I'll show you some pictures of that, and Finnian will talk a little bit about his experience. That leukapheresis product at the moment is shipped to the manufacturing facility, which for the commercially manufactured products is overseas, and for our own in-house experimental products is here at Peter Mac. The cells are transduced with a virus, and the virus contains the required elements that enable expression of the CAR T receptor on the surface of the CAR T cells. And there are different types of viruses that can do that. Manufacturing of those viruses obviously is complex and expensive, and there are other non-virus ways of achieving this, but at the moment most products have their genetic transduction occur through the use of virus. Those T cells are then expanded in the lab. There's several washing steps to remove the residual virus, and then you have a product. And the product comes back to the patient in a bag like this. And often the patient's disappointed to discover that the product is 11 mils in a single bag and it goes in 15 minutes, and they say, what, that's my $500,000, or that someone's paid that much money for this. And that's what it looks like. And that occurs at the treatment site. I'm going to skip over manufacturing. Here at Peter Mac, this is our workflow, and our workflow includes all of those steps. But the key thing I wanted to emphasise here is that prior to infusion, there's lymphodepleting chemotherapy. So we usually give fludarabine and cyclophosphamide to ablate the patient's lymphocyte population, but particularly to ablate the regulatory T cells that can prevent T cell expansion. The infusion day happens here, and typically we keep patients for some products as outpatients, while some patients will be admitted to the ward where they're managed on the ward. And then we have a system for monitoring patients very closely, usually daily reviews every day as an outpatient, and then we admit patients if they develop the side effects of CAR-Ts. So I'm going to turn now to mature B-cell neoplasms and talk specifically about diffuse large B-cell lymphoma. Diffuse large B-cell lymphoma is one of the most common lymphomas. It's diagnosed at a median age of 65, and it's somewhere between, it forms the basis of many a haematologist practice because it's a relatively common disease. Now, our approach with diffuse large B-cell lymphoma is to use conventional chemotherapy, so R-CHOP chemotherapy, rituximab, the anti-CD20 antibody, in combination with CHOP chemotherapy, which is given for 6 cycles. That's our standard backbone treatment, and it delivers cure in around 2/3 of patients. And so most patients who have this diagnosis go on to be cured after frontline chemotherapy. If a patient has refractory or relapsed disease, then we think about it in terms of whether or not they're fit to receive intensive chemotherapy. And here I'm talking about high-dose chemotherapy with stem cell rescue and what we call autologous stem cell transplantation. In this particular procedure, we use an apheresis procedure to collect a patient's hematopoietic stem cells, keep them to the side, give high-dose chemotherapy which has the side effect of ablating the marrow, and then give the stem cells back to rescue the marrow from the toxicity of chemotherapy. The main purpose of that procedure is to deliver high-dose chemotherapy. The stem cells themselves don't deliver any kind of disease control. But that technology is used to enable us to give these high doses. And that can cure, people say, around 50%, probably more like 30 to 40% of patients will be cured with high-dose chemotherapy and autologous stem cell transplantation. If a patient relapses after that, though, we don't have any standard therapies available to us at the moment, apart from chemotherapy, which is usually given with palliative intention and which is unlikely to deliver cure. So that's the current treatment paradigm for diffuse large B-cell lymphoma. And we think somewhere between 200 and 400 patients sit in this group here every year in the Australian population, although estimates of that number vary significantly. What we don't know very well is how those patients actually go. Most haematologists you speak to will tell you that diffuse large B-cell lymphoma after relapse, after a stem cell transplant, or in patients where you can't get stem cell transplantation, has a dire prognosis, but it's actually very hard to find good data that supports that argument. And so in this aggregated study looking at 600 patients with relapsed and refractory lymphoma, you can see that patients who went on to receive stem cell transplant did okay, but patients who didn't receive stem cell transplant did particularly poorly, or patients who relapsed after stem cell transplant had a long-term outcome sitting somewhere between 10 and 20% survival over the observed period. So along came this trial using CAR T-cells. And the first trial was the Juliet study using a drug which is now called, or I shouldn't call it a drug, but a cell product called Tisagen Leclucel. It's also called Kymriah, and it's now made by Novartis. And it was developed at the University of Pennsylvania, a CAR T product with a 4-1BB co-stimulatory domain and targeting CD19. And this trial was run in America, Europe, and Australia, as well as Japan, and the design of the study was as a single-arm study. There was no randomisation. Patients were screened, had their apheresis and cryopreservation performed, and then enrolled in the clinical trial and received bridging chemotherapy to hold the lymphoma during manufacturing. Those manufacturing steps that I've talked to you about can take a minimum of 2 weeks, but in this clinical trial took 3 or 4 months. And so holding an aggressive lymphoma during this time is extremely clinically challenging, particularly when you're talking about patients who've already demonstrated refractoriness to current available therapies. And so our goal in that time during bridging chemotherapy is to maintain patient fitness and not cause excessive toxicity from what is otherwise therapy that's not going to deliver effective long-term disease control. Patients were then given lymphodepleting chemotherapy with fludarabine and cyclophosphamide and had their product reinfused and then were imaged for follow-up. There's another product that's currently on the market and approved in the United States, which has been assessed in a trial called Zuma-1. This is made by a company called Kite, which was recently purchased by Gilead and came out of the NIH program led by Steve Rosenberg. Same target, same CD3 zeta chain, but a different co-stimulatory domain. Signal 2 was delivered by CD28. And in this trial, the design was about the same. Patient numbers were fairly similar, and they recruited different subtypes. Primary mediastinal B-cell lymphoma and transformed follicular was addressed in a special subgroup. And again, screening leukapheresis. What was different about this trial is patients were not allowed to receive bridging chemotherapy. So they only went onto the trial if a doctor thought they could live long enough to receive the CAR-Ts without any additional therapy. Where this trial outperformed the other trial was manufacturing was generally not longer than 21 days as opposed to 3 months. This is manufactured on the West Coast US and Chimeric on the East Coast US. And broadly speaking, these are the outcomes of the trial. Juliet study by Novartis screened 238 patients, enrolled 167, but lost quite a few patients due to manufacturing or related issues, death during bridging chemotherapy. Some patients withdrew and their reasons for withdrawal weren't defined, and only 115 patients got infused. Whereas with the Zuma-1 study, 111 were enrolled, there was no bridging therapy given, and 101 patients were infused. So less patient selection within the protocol, but possibly a step of selection before patients were screened in the clinical trial. And what we saw here is that overall, about 40% of patients had a durable complete remission over the various time points that the trial data has come out. So we saw that at around 6 months and 12 months, about 40% of patients are cured with each study. And what's striking is that where patients were observed to have responses, particularly when they got out to around the 12-month period, if they had a response, they seemed to be keeping the responses for those patients who've been observed beyond this period. And this data has been updated recently, and we see these durable complete responses occurring, sitting at around the 40% rate for both products. And so that seems to very significantly outperform what we see with standard of care treatment. What are the side effects of CAR-T? Well, there are 2 key side effects that we think about. One is the cytokine release syndrome. As T cells see their target and expand, they release a range of cytokines, and the cytokine release syndrome is a consequence of multiple cytokines, but predominantly IL-6. And it manifests as essentially a sepsis-like syndrome. Most patients who receive CAR-Ts develop fever, and then some go on to develop other features that we would typically associate with sepsis, but obviously without an infection driving it in most cases. So fever, moving on to hypotension requiring vasopressors, and sometimes moving on to fully blown shock with multi-organ dysfunction and respiratory dysfunction. And our approach to managing these toxicities is initially with supportive fluids and supportive care, subsequently with an anti-IL-6 agent, tocilizumab, which is now routinely available, and its use has sort of evolved over the course of these clinical trials, and then also dexamethasone and other higher dose steroids as the complications become more severe. The second very significant toxicity is called the immune effector cell neurotoxicity syndrome. It's called a syndrome because people don't really know why. A little bit of it might be about IL-6, probably IL-1 is important in this. And here a patient can develop confusion, inability to name objects, inability to use words properly, often early symptoms, difficulty handwriting, mobility problems. But there have also been patients who've developed severe encephalopathy within hours of the infusion, and patients who have died from cerebral edema using this technology. And there have been patients who've been kept alive through neurosurgery to decompress the brain after these side effects. So this can be a very, very rapid syndrome, and occurs differentially depending on what the co-stimulatory domain is with the CAR-Ts. We learned from the clinical trials that the peak expansion for CAR-Ts with the Zuma trial occurs around day 7, and we saw that both the area under the curve for CAR-T expansion and also the peak levels correlate somewhat with objective response rate, also with the presence of neurologic events, but less so with the presence of cytokine release, which means if a patient's been infused and they don't get cytokine release, it doesn't mean they won't respond. And if they do get cytokine release, it doesn't mean they will respond. So I'm now going to ask Finnian and Bernie to come and talk a little bit about experience of being on the Juliet trial, which is the Novartis study, and they've come all the way from Sydney just for this, so I'm incredibly honoured and grateful for this. I guess I'll start by grabbing a microphone for me, turning it on, and I'll say, can you tell me a little bit, Finnian, about the diagnosis of your lymphoma, how you presented and what it was like being diagnosed, and what treatments you were initially given in Sydney?
So obviously it's 4 years ago now since I was initially diagnosed, and the memory's not the best, but we'll work with it. So I was initially diagnosed in January. I had my lymph nodes in my stomach were all swollen. I had a 200 mil tumour at my stomach, and I could feel it, and it was pressing against my bowel. I was starting to have problems. When they brought me in, I went in on a Saturday. They were going to operate straight away. They didn't know what to do. Our hematologist arrived in on Monday, and I started on RCHOP on the Friday of that week. Then we did the biopsies probably on the Monday or Tuesday, and the results came back probably about 7 or 8 days in. So just after the first RCHOP, and it said we'd a double hit, diffuse large B-cell lymphoma.
What were you told about double hit lymphoma at the time?
I think the doctor didn't want to frighten me too much, so she said, look, I think we need to change the treatment we're on. So she says, at the moment, she says, it's up to you, but obviously we can stay on RCHOP, but we find that, and they hadn't a lot of experience in the double hit at the time, like saying, you know, the sort of the response rates. And they said RCHOP is good, but we feel that EPOCH was the one we went on, would be better. Now we had to do a second run of RCHOP, so I got 2 RCHOP and then changed to EPOCH for the following 4. And that was 5 days in hospital, I think fortnightly.
For 6 cycles?
For 6 cycles, yeah.
And did that put you into a complete remission?
Not at all. So because it was 200 mil in size, it shrank probably around 80 mil. So it got right down. From there then, we sort of said, right, let's do an auto. Yeah, it was an auto we were going to do.
So autologous transplantation.
So we went...
Salvage chemotherapy, stem cell collection.
That's correct.
And then moving to high-dose chemotherapy with autologous stem cell rescue, which is the standard therapy for patients with relapsed disease.
Yep.
And you've described double hit lymphoma, which we now recognise as being one that's, you know, more likely to be refractory to chemotherapy.
That's right.
Yep. So on that then, we went and, again, spent 3 weeks in hospital for the chemotherapy. So we got that. We went and did the auto, the stem cell transplant. And we did another scan, so a PET scan after that. I think we were left with 13 mil or something. It had shrank a bit, but not enough. And what we found then, it actually had spread. So there was a couple of little lumps. One came up in my arm, other parts of my body, things like that. And that's when they sort of said, look, it's not looking good. You know, we'll reach out to other people and see what other treatments are available. So that started from there. That was probably July 2015.
That's when we got introduced to you.
Right. So there's this. so there's an app that was used apparently to find the trial, something that's currently available for patients looking for clinical trials. That was actually developed by Judith Trotman in Concord, who I think you know.
Yes.
And they used that app to find the trial and gave us a call. But at that stage we had a problem, didn't we? Because you had active lymphoma. We knew this trial was available. But we also knew that manufacturing would take some time.
That's correct.
So what we did as well, we actually were trying for an allogeneic now. So my brother. So we tested. Obviously, family. I have 4 siblings, and one of them was a match. He actually lived in Ireland, but we had got the Red Cross to do a match, and he was preparing like if there was a possibility, and we had met with Chris O'Brien Lifehouse, and they said, look, if we could get it to sort of write down, we'd chance that, maybe have a go at that. But that was, we just weren't getting remission at all. They wanted remission, but even they said if we could get it right down with as much chemotherapy as we could, we would have a go at an allogeneic.
So they said to take you to transplant, to allogeneic transplantation, you needed to have a remission first.
Yes.
And what was the reason? Did they explain that?
Because of the chemotherapy that I would get, it would obviously spread. The cancer would spread throughout my body because my immune system would be knocked out.
Yeah.
And by the time I would get my brother's stem cells, they wouldn't, like, the cancer would have taken over. So I needed remission.
So that time it takes for the allogeneic response to build up, in that time you can't have other anti-cancer therapies, and you've just got to hope that the immune response beats the lymphoma.
Yeah.
And with these aggressive, fast-moving lymphomas, that's less likely. So they ask you to be in a complete remission at the time you get an allotransplant.
So then we decided from Sydney as well to go with radiation. So it was lenalidomide and radiation together to try for that. And we still had a collection of my stem cells.
And that's experimental, really.
Yes,
isn't
totally.
it? Reaching
Totally.
for anything off the shelf, we would still consider that to be
It was
experimental.
basically just trying to keep him going
Yeah.
until perhaps a trial became available.
Yeah.
Because it looked very bleak at the time. There didn't seem to be anything. Oh,
So
you didn't look bleak.
So and we did reach out to overseas as well.
Yeah.
So we did actually get a response from yourselves, from Peter Mac, and from, I don't know what the company name is, but for a drug called Selinexor. So we actually got a response, and I got that on compassionate grounds. So when we came and spoke to Michael, so we saw Michael here, the trial wasn't opening until, was it January or something?
Right.
And this
We were
was
moving
probably
hospitals at the time,
August.
weren't we?
This was August, and I remember you saying that you can't use Selinexor and radiation together, because we had a chance of getting Selinexor at that stage, and they said don't use it together because it's never been tried before.
So selinexor is a SINE inhibitor, so selective inhibitor of nuclear export, which has not come to market in lymphoma and has recently come to market for myeloma in the US, with some pretty marginal activity data for those of you who are interested.
I found Selinexor to be
Tough.
quite a tough drug. It made him quite volatile. His mood swings. He was going a bit nuts. So.
But in fairness, he'd been knocked around a bit, hadn't he?
Yeah.
Been knocked around a bit.
But it was the last one before the Juliet trial. So, yeah, but like because of radiation and lenalidomide did not work. So then we were hitting September, October, and Selinexor came into play. So I had to use lenalidomide for 6 weeks because that was the only way we would know if you got any response at all. So we had to just go with that. In the meantime, radiation actually helped, but they said radiation in lymphoma is not common. So
Well, it's a very good anti-lymphoma treatment, but in large cell lymphoma, unless you can capture everything within the radiation field, and even then it tends not to be curative as a single treatment, but to hold or to put someone in a remission can be effective. So then you came and saw us, and we said, let's do this trial, and we decided to collect the lymphocytes, do the apheresis procedure.
So we had a rough journey, as you know. You obviously know better than anybody. But so in January when we came down, what we discovered was my bone marrow came back aplastic. So that meant my bone marrow had died, basically. And we think maybe the Selinexor and all the different treatments definitely didn't help it at all. From there then it took, I think, about 8 to 10 weeks for the bone marrow to recover very slightly. And there was something in the trial as well that changed, because I couldn't make the criteria. So that we didn't get the collection until March. So from when we initially spoke, it was something like 8 months. So we had to stay alive for 8 months.
We did think that the collection date was on St. Patrick's Day.
Yeah.
And that in itself was
A good sign.
A good omen.
Good
yeah.
sign. Yeah.
So, and then we made it in one day. You
Yes.
actually booked us for, I think, 4 days, 3 or 4 days, and we got a full collection on
On that day.
Yeah.
So you then gave us some chemo to hold us as well.
Yeah.
And
Which you'd struggled with, if I remember
very,
correctly.
yes, I remember you said it was even in older people, this was very easy and we should tolerate it. And I think I got 1 dose and that was it.
Yeah. Amazingly, over all the treatments, that particular chemo was the one that stuffed him around so much.
I thought you were going to say it was
But
it
it seemed, obviously, it must have done something, but it was the one that upset him the most.
Yeah. I think by this time you were very, very heavily treated. So we had this practical challenge of holding someone's disease while the CAR Ts are being manufactured. Now, Novartis have changed what they do with manufacturing, and the manufacturing time is shortened, but this is one of the challenges with CAR Ts. One of the differences with allogeneic transplantation is the CAR Ts can treat manifest lymphoma, and the T-cell expansion and the anti-lymphoma effect is sufficient to put patients into a remission. Whereas with allogeneic transplantation, waiting for that expansion and response, you have this competition for these the aggressiveness of the disease. So ultimately, CAR Ts were reinfused.
Yes.
That's correct. But with the apheresis, that was on March. And then we came back and we got it reinfused on the 14th of July. And that's 3 years. No, it's 2016.
2 weeks ago, 3 years ago.
3 years ago.
So,
And what you said earlier is very true. It was nearly like an anticlimax. The amount, I think it was 19 mil.
maybe it wasn't
Yeah.
a lot. And
And it was like, is that it? Like, this is unbelievable.
it didn't even take an hour, did it? It wasn't long.
Yeah.
Yeah.
And we were lucky enough to be outpatients. We were down in Melbourne on and off for about 8 weeks altogether. But we were allowed to travel initially after the first
After
2
the
weeks.
first couple of weeks.
Yeah, so it went surprisingly smoothly, and I don't know about Finn, but at the time I was thinking, this is too good to be true. He's well, he had no side effects, and, it was fantastic.
Yeah, no, we tolerated that very well. Like there was, I remember everybody said you'd have cold, like flu-like symptoms for 10 days. And we didn't have any. I think we might have called up every day or every second day, but it was like a half an hour in and out and off again. So we were very lucky. I think, we didn't have any of those symptoms at all. And then the first scan we did after, I think was September, and we actually had positive action, or it shrank, I think, from,
sorry.
I think I remember, was it 13 mil or something it shrank to? It was something huge, like from 30 mil, like it went down in size.
It's fair to say every scan was an anxious moment,
Oh,
absolutely.
as it is for all of our cancer patients, I guess. And, so pretty plain sailing, no fevers, no confusion.
No.
Did you notice any confusion?
Oh, he's always
confused. So no one could tell.
Yeah, but we were nearly on the, thinking, should you be sick?
Yeah.
We're nearly wishing that he was feeling a little bit
yeah.
sick, so then it might be working, not realising that obviously it was working and doing a fantastic job.
Yeah.
It was scary times because he was so well, almost.
Yeah. So you're certainly an example of someone who sailed through it in some ways, but we had to string things out in terms of disease control.
Yes. Yeah.
So the other point, I suppose, and you made earlier, was we didn't get the preconditioning chemo either.
Yeah. And the reason was we'd flogged you with so much other chemo, you actually didn't need it at that moment. But it was a bit hairy because there's very clear evidence or reasonably clear evidence that the preconditioning chemo makes a difference to CAR T-cell expansion and engraftment. And there are CAR T-cells that are now available in clinical trials which completely didn't work without conditioning treatment. But because of how your lymphocytes were from all the other treatments, we'd pretty much given you that with all the other treatments anyway.
Yep.
So we didn't want to derail things by making you sicker.
So I must say our checkup in Melbourne, down here, Peter Mac, in the January 17 was probably one of our scariest. We were very anxious all over Christmas because he was so well, almost. It was like every day, are you okay? Are you feeling sick? And then just to get such great news. You're the bearer of great news every time we come down.
So 3 years. So 3 years now. So that's amazing. And I think in terms of the natural history of diffuse large B-cell lymphoma, we can say with confidence that CAR T's modified things in your case. Does anyone have any questions for Finnian and Bernie at the moment? There's time at the end to come back and ask.
I'm very curious what kind of diet you take and help you to improve your health.
The question is about diet.
So, yeah.
Actually, can I just, at the beginning, Finnian started doing naturopath stuff. He was taking, when he was first diagnosed. But in fact, in retrospect, when we look back, we think now that although it made him feel very well, it might have been masking, the symptoms where he was actually worse internally, but he appeared to be well. So, but we decided not really to change diet, because we had seen a friend of Finnian's go through extreme diet change and didn't recover. In fact, he got worse. So we decided that we'd kind of stick with our diet, which is reasonably good anyway.
And it still hasn't changed now. It's
Yes.
eating the exact same.
Yes.
But again, to say about that naturopath, I think it's good, but the problem with it, I was able to take more abuse, or my body was able to take more abuse, because I built myself up so much, I think. And like looking back, if I didn't take it, maybe I wouldn't have taken as much of abuse. I don't know.
Okay, well, we might keep going and then we'll come back
Yep.
for other questions. Is that okay?
All right.
Oh, you've got one more picture.
Yeah. So we thought we'd never reach the milestone of the 50th.
All
and we
right.
did. So, yeah, that was it. Great.
Fantastic.
Thank you.
Thanks,
Michael.
Michael.
Thank you. So these are Finnian's slides, and they show relatively low volume, but nevertheless clinically significant lymphoma here pre-CAR Ts with complete remissions sustained at 6 and 9 months. This is another patient who we treated in the Juliet study, a man who had relapsed and refractory diffuse large B-cell lymphoma. He presented with R-CHOP. Was treated with R-CHOP, had severe complications from R-CHOP, but then was shown to be refractory with predominantly extranodal disease, went on to receive R-ICE chemotherapy, which is our standard intensive salvage chemotherapy, and completely progressed through that with absolutely no response, and was told to go home and prepare for the worst. He went back to his country town in northern Victoria and tried palliative radiotherapy. As you can see, he had very unusual extranodal lymphoma involving his skin and his bones. He had a golf ball-like skull with big defects in his skull that you could see on view, big lumps. It was easy to get biopsies to prove that it was lymphoma. And we entered him. He was told he had no treatment options, but then came back a couple of months later for the Juliet study, having had radiation to all the symptomatic sites that his poor radiation oncologist could target. We treated him then with some different conventional chemotherapy, not with the expectation of a response, but with the hope that it would hold him within the Juliet study. We collected his lymphocytes. He broke 3 bones from his lymphoma during the manufacturing period and had to have a number of fixation procedures at St Vincent's Hospital. He had infection and other hospitalizations, but we gave him the CAR Ts, and he's also in a remission almost 3 years later. So quite remarkable, totally chemo-refractory patient responding to an immunotherapy. And he did require bridging chemotherapy, and we don't know whether bridging chemotherapy augments or harms these responses. It's still a research question. So what's the treatment paradigm now? We've got this clinical trial, single pivotal clinical trial with Kymriah, another clinical trial with the Gilead product. We've seen around 40% durable complete remissions, no randomisation yet to show that it's better than chemotherapy, but any physician in the space who've looked after patients such as Finnian, such as the other patient that I presented, feels that there was no other treatment that could have delivered this outcome. And so on the basis of approvals in the EU and Canada, the United States, so there's around 10 countries now where there's approval and government funding. The current standard of care for patients who fail autologous stem cell transplantation or 2 lines of chemotherapy for diffuse large B-cell lymphoma, if they're CAR T fit, is to receive CAR Ts, and hopefully a good proportion will go on for cure. Now, in Australia, at the moment, the only approval for CAR T is acute lymphoblastic leukemia in the paediatric and young adult population. The reason this trial was done in this age is because patients died from cytokine release syndrome who were older than the age of 25 when they received the full dose of CAR T. So Novartis, in collaboration with the university that invented this product, decided to start with younger patients who had more to gain from the treatment. And here we're talking about a relapsed acute lymphoblastic leukemia, the most common cancer in children, where most patients are cured by chemotherapy. Some patients receive allogeneic transplantation frontline if they have high-risk disease, and allotransplantation is effective in relapsed or refractory disease. But in patients who relapse after their allotransplant, the outcomes are just dreadful. And even these high-tech CD19, CD3 bispecifics, which deliver significant advantage over the short term, do not deliver cure. And so our expensive high-cost drugs are offering nothing other than a bridge to something else for this patient population. And here, this is the same disease as Emily Whitehead. Kymriah, the Novartis product, histogenlecleucel, delivered an 80% overall response, and around 2/3 remain alive several years after treatment in the clinical trial. So a remarkable outcome that led ultimately to the federal government funding CAR Ts for acute lymphoblastic leukemia in this population. And this is the population that we're currently treating with government funding here at Peter Mac and at the Children's Hospital down the road. We're waiting on outcomes for diffuse large B-cell lymphoma. But our approach with the observation of the single-arm studies here at Peter Mac is to try to integrate CAR T-cells into what we do. And we've identified that patients with lymphomas like Finnian's, these double hit lymphomas, or patients with high clinical risk factors who also have an interim PET scan which is positive, have a pretty poor outcome when we use conventional chemotherapy and autologous stem cell transplant. And so for those patients, we now have an investigational single-arm phase 2 study where they just receive CAR T without any chemotherapy in a single-arm study. So highly experimental, but fairly tempting for those highest risk patients. For patients who relapse within 12 months or have primary refractory disease, there are now 3 randomised clinical trials randomising patients to receive either autologous stem cell transplantation or CAR T. And then we have a number of clinical trials looking at patients who relapse outside of that 12-month high-risk threshold. There's still a role for autologous stem cell transplantation in that lower risk relapse group as far as we know. And we're hoping that the government and the companies will come to an agreement whereby we can access CAR Ts for patients like Finnian in his particular situation outside of the context of clinical trials. But this is very expensive treatment. The US market price is around 500,000 Australian dollars per dose, not including the costs of the ICU stays, the cognitive complications, the use of immunoglobulin, which we didn't touch on. Finnian takes immunoglobulin monthly to deal with recurrent respiratory tract infections. So addressing those costs of care become a real challenge in a socialised healthcare environment like ours. And so hopefully, that's currently before the committees with government at the moment. We've got about 5 minutes left, so there's time for questions. Before I do, I just wanted to mention briefly about setting up a service here at Peter Mac for standard of care CAR Ts and talk a little bit about the depth of history here. Firstly, on the left is Donald Thomas, the person who discovered and developed allogeneic transplantation with Jeff Scher, who was one of the first people in Victoria to do allogeneic stem cell transplant, and is, of course, was the leader of the Allogeneic Transplant Service until relatively recently at Royal Melbourne Hospital. So Jeff, of course, pioneered cellular-based therapies for hematologic malignancy here in Victoria. Mike Kershaw, Joe Trapani, both here in the research division of Peter Mac, have invented a CAR T. They were the first in the world to deliver CAR Ts, the Lewis Y CAR T, which is still in clinical trials for solid tumours here at Peter Mac. The hospital itself has taken a very aggressive development approach, identifying that CAR Ts are an area for future use in cancer. And even back in the old East Melbourne site, we had clean rooms that were set up through philanthropy to actually manufacture these CAR Ts on site. And now here we have 10 clean rooms, which we're hoping with government grants to double to 20 clean rooms so that we can manufacture commercial and experimental CAR Ts here on site using appropriate good manufacturing practice that allows safe delivery of these treatments to patients. This is all the CAR T trials that are going on in the US at the moment, more than 60 around the world, more than 60 targets across a range of cancers. And so we think that this really will be an expanding area for cancer care. So in summary, CAR Ts are antigen specific. They can effectively kill cells independently of MHC molecules. Clinical responses have led to commercial approvals, but there are important safety risks. Not every patient has an event that makes the CAR Ts unsafe. We need to keep patients close to the CAR T treatment centre. But many patients will have an experience like Finnian's where it's a very positive experience compared to conventional therapies. But we want to have better than a 40% long-term outcome. We want less toxicity overall, and we want to see new indications. And I think probably the next indication will be in myeloma, as we heard last night on the news. So these are my acknowledgements, and thanks very much.
Amazing presentation. Thanks very much, Michael, Finnian, Bernie. I might actually invite you back up to the stage just so that we can answer any questions that the audience have made for any of you. And so I'll just quickly kick it off, both to either of yourself, Finnian and Bernie. When you came to the point of receiving CAR T therapy, what was your mindset at the time of, when you went into it? Were you thinking this was a real Hail Mary, or did you always sort of believe things were going to work out? I just kind of wanted a bit of a snapshot of how you were feeling at the moment that you got onto the trial.
I actually, personally, I thought it'd work.
You
Finnian never had a doubt. Yes,
I
never
believed
had a
it would
doubt.
work. I was actually, when I couldn't get onto it, I was very upset.
Yep.
I actually spoke to one of my doctors in Sydney and was trying to manipulate my own blood to get onto it. I just wanted to get onto it.
Right.
But there was only, I think, during the whole 1.5 to 2 years of Finnian's treatment, there was only one moment when he said he was going to give up. And 5 minutes later, it was like, nope, all good.
Was that anything to do with you, Benny? Or was that...
No, not at all. It was all mouth ulcers and just pain and, horrendous times.
In turning his attitude around, but anyway, yeah, all right.
Yeah, no, it's always been, Finnian's always been extremely positive, and I think that's possibly one of the reasons he got through it. His positivity never really faltered whatsoever.
No, I think when we met, what gave me confidence was the old Peter Mac. We got into the old Peter Mac hospital and I felt really reassured. I don't know what it was. I just liked it and I thought, we're going to get on this thing no matter how. And that was my goal. So when you have a goal, I think you just aim for that goal, and that was it.
And you've also got that, it's a life or death. There's no choice. You choose the trial to live or, or death is the only other option.
We didn't have a choice.
Look how great it worked out.
Yes, absolutely.
Any other questions on the answer, Michael or Finnian or Bernie?
Yeah, could you tell me what the symptoms you had leading up to it were?
So probably a couple of months earlier, I felt little lumps in my tummy and I should have went to the doctor. But I even laughed because I thought they were moving. So what it was, different lymph nodes were swelling up. Probably for about 5 or 6 weeks, but it wasn't sore. I didn't have any pain or anything. Just at Christmas, I got an awful pain in my stomach, and I was at that stage. I think I was just finding it hard to go to the bathroom, actually. So I was just pushing against all my organs.
And did you go to a local doctor?
Yeah.
And he picked it up?
No, he said an ultrasound. So it took me 3 or 4 days to get the ultrasound.
You were lucky because I frigged about with doctors for 18 months before I got a doctor that said, I want to see you, send you for a CT scan. And then they picked it up.
I'm also very pushy.
So when I actually went for the ultrasound, the funny thing was, and I didn't cop on myself, they came in and actually had a bit of a chat. So the nurse was there and a doctor came in, so they called the doctor. And they said, I think we need to do a CT. And that was totally, like, you're here now, we get to do one. So I should have copped there was something wrong then.
He also did have night sweats, and he was always tired. He'd come home from work and he'd just sleep but he would be he wouldn't agree with me on this, but because I'm going through a stage in life as well, he blamed the night sweats on me, whereas I knew it was him. So there were symptoms when we do look back, there were symptoms, but we kind of overlooked them, really.
Hi, Finnian. Just a quick one. Did I hear correct that you searched online and found an app to find the clinical trial, or?
Michael will help with that one. So it was our doctor in Sydney that actually searched.
Yeah, so there's an app called Clinical Trial Refer, which is on the Google and Apple stores, and it was actually developed by someone in the hospital where Finnian was initially treated, and it's won a number of awards internationally for improving cross referrals, and that includes the inclusion criteria and those sorts of things.
We also did a bit of Dr. Google, and I got in contact with a German clinic and also a Pennsylvanian professor but they both agreed when I sent Finnian's bloods over that he was too ill to travel. So to find something more local, and that's when Michael and our Dr. Jane in Sydney was amazing.
And in saying that, it was Concord. They were definitely ahead of it. And they support Peter Mac completely, because I was told by people in Sydney that when I got the opportunity to come here to take it, I could have actually transferred back to Chris O'Brien, and I think I'm very comfortable here. Again, it's with the people you're with. So for me, this was the one that, and I was advised by everybody, and our hospital in Concord was brilliant for that.
I'm wondering whether the patient yourself worry about a relapse, and how about the professors and doctors explain what contribute to relapse?
So for me, I actually, funnily enough, don't think I'll ever relapse. So I'm very confident. I actually thought because my T-cells are infected that they'll always be infected. So I don't know. But I presume that, and I think they're working away all the time.
So I think Finnian's actually touched on a couple of things there. The first thing is that we think that relapse is linked to the persistence of the T-cells. To some extent. So to some extent, it's the depth of the initial remission is really important. For acute leukemia, how long the T-cells hang around for is probably important as well. And we can actually test for that. And in fact, Finnian has been repeatedly tested for that. The other thing, one way of testing for that is to look at the health of normal B cells. And one measure of that is the ability to produce antibodies. And we know that Finnian isn't very good at producing antibodies because he gets colds and coughs and chest infections, and we need to give him donor antibodies to prevent that complication. And that's a side effect of normal CD19 positive B cells being hit by the CAR-Ts. And the fact that that's persisted is probably a sign of persistence of the CAR-Ts. Now, with leukemia, more so than lymphoma, we know that some patients relapse because they lose CD19 on the surface of the leukemia cells. So that's one potential mechanism of relapse that we understand.
Right. And with that as well, you're saying about the treatment, it was probably 9 or 10 months or a year before I actually started receiving that. So and it was a flu symptom that I had all the time.
All right. So join me again in thanking Finnian, Bernie and Michael. Thank you very much, guys.